General Biology, Part 03. Platelets. Platelets are essential for blood clotting. When a blood vessel is injured, platelets clump together to form a plug that stops the bleeding. Platelets also release chemicals that help to activate the clotting cascade, which is a series of reactions that leads to the formation of a blood clot. Platelets are the third type of blood cells, after red blood cells and white blood cells. They are also called thrombocytes. Platelets are small, colorless cell fragments that help blood clot. Platelets are made up of proteins, lipids, and carbohydrates. The proteins in platelets are essential for their function in blood clotting. When a blood vessel is injured, platelets release chemicals that attract other platelets to the site of the injury. The platelets then form a mesh that traps blood cells and helps to stop the bleeding. The clot that forms when blood clots is called a fibrin clot. The fibrin clot is made up of a protein called fibrinogen. Fibrinogen is converted into fibrin, which forms the mesh that traps blood cells. Blood clotting is a complex process that involves many different factors. Platelets are just one of the factors involved in blood clotting. Other factors include proteins, blood vessels, and the nervous system. Blood groups. The blood groups are determined by the presence or absence of certain antigens on the surface of red blood cells. The most common blood groups are A, B, AB, and O. Antigens. These are proteins or other substances that are found on the surface of cells. Antibodies. These are proteins that are produced by the immune system to fight against foreign substances. When a person receives a blood transfusion, the blood must be compatible with their own blood type. This is because the antibodies in the recipient's blood will attack the antigens on the donor's blood cells if they are incompatible. For example, a person with blood type A has A antigens on their red blood cells. They can only receive blood from a donor with blood type A or O. If they receive blood from a donor with blood type B, their antibodies will attack the B antigens on the donor's blood cells and cause a reaction. Agglutination is the clumping of red blood cells. When blood from two different blood groups is mixed, the antigens on the red blood cells of one person will react with the antibodies in the plasma of the other person. This will cause the red blood cells to clump together. The agglutination reaction is important because it can help to prevent blood transfusions from being mismatched. If blood is mismatched, the red blood cells of the recipient can clump together and cause a serious reaction. 3. The Rh factor. The Rh factor is a protein that is found on the surface of red blood cells. People who have the Rh factor are Rh positive, while people who do not have the Rh factor are Rh negative. About 85% of people are Rh positive. The Rh factor is not important for blood type, but it is important for blood transfusions and pregnancy. When a person with Rh negative blood receives blood from a donor who is Rh positive, their body will produce antibodies against the Rh factor. These antibodies can attack the red blood cells of the baby if the mother is Rh negative and the father is Rh positive. This can lead to a condition called hemolytic disease of the newborn, HDN. HDN is a serious condition that can cause the death of the baby. There are a few ways to prevent HDN. One way is to give the mother Rh immunoglobulin, RhoGAM, during her pregnancy. RhoGAM is a medication that helps to prevent the mother's body from producing antibodies against the Rh factor. Another way to prevent HDN is to give the baby Rh immunoglobulin after birth. This will help to prevent the baby's body from producing antibodies against the Rh factor. Blood group matching table. Blood group is inherited from parents in a Mendelian fashion. This means that each parent passes on one of their two blood group alleles to the child. The child's blood type is determined by the combination of the alleles they inherit from their parents. Table shows the possible blood types of a child based on the blood types of the parents. 4. For example, if the mother has blood type A and the father has blood type O, the child can have blood type A or O. This is because the mother can pass on either her allele or her O allele to the child, and the father can only pass on his O allele to the child. The table is divided into 4 sections, one for each blood type, A, B, AB, and O. Each section shows the possible blood types of the child if the mother and father have the blood types listed at the top of the section. The table is a useful tool for predicting the possible blood types of a child. However, it is important to remember that the table is not always accurate. There is a small chance that the child's blood type will be different from what is predicted by the table. Punnett square. A Punnett square is a diagram that is used to predict the possible outcomes of a genetic cross. A genetic cross is a mating between two individuals. The image shows a Punnett square for a genetic cross between a person with blood type A and a person with blood type O. The blood group alleles are represented by the letters A and O. The top row of the Punnett square shows the possible alleles that the mother could pass on to the child. The left column shows the possible alleles that the father could pass on to the child. The cells in the Punnett square show the possible combinations of alleles that the child could inherit. The child could have blood type A, blood type O, or blood type AB. The probability of the child having each blood type can be calculated by looking at the number of cells in each row or column. For example, there are 2 cells in the row with 2 alleles. This means that there is a 2/4 chance of the child inheriting 2 alleles and having blood type A. There is 1 cell in the column with 1 allele and 1 O allele. This means that there is a 1/4 chance of the child inheriting 1 allele and 1 O allele and having blood type AB. The Punnett square is a useful tool for predicting the possible blood types of a child. However, it is important to remember that the Punnett square is not always accurate. There is a small chance that the child's blood type will be different from what is predicted by the Punnett square. Here are some other things to keep in mind about Punnett squares. The Punnett square is based on the principle of Mendelian inheritance. This means that each parent passes on one of their 2 alleles to the child. The child's blood type is determined by the combination of the alleles they inherit from their parents. The Punnett square can be used to predict the possible outcomes of any genetic cross, not just blood types. The Punnett square is a useful tool for understanding genetics, but it is not always accurate. There is always a small chance that the child's phenotype will be different from what is predicted by the Punnett square. 5. Q. A married couple adopted a male child. A few years later, 2 boys were born to them. The blood group of the couple is AB positive and O negative. The blood group of the 3 sons is A positive, B positive, and O positive. The blood group of the adopted son is A, O positive, B, A positive, C, B positive, D, cannot be determined on the basis of the given data. 2011. Taxonomy. Taxonomy is the science of classifying organisms. It is based on the principle of hierarchical classification, which means that organisms are classified into groups based on their similarities. The biological classification system has 7 levels, from the most general to the most specific. 1. Kingdom. The next level of classification. There are 5 kingdoms: Animalia, Plantae, Fungi, Protista, and Monera. 2. Phylum. The next level of classification. There are 35 phyla in the Animalia kingdom, for example. 3. Class. The next level of classification. There are 13 classes in the Mammalia class, for example. 4. Order. The next level of classification. There are 19 orders in the Primates order, for example. 5. Family. The next level of classification. There are 3 families in the Hominidae family, for example. 6. Genus. The next level of classification. There are 2 genera in the Homo genus, for example. 7. Species. The most specific level of classification. There are 2 species in the Homo sapiens species, for example. Classification of organisms. 5-kingdom classification of living organisms by R.H. Whittaker in 1969. 6.